In this video, we'll take a detailed look at the library design process. By the end, you'll be able to describe the key stages of library design, identify the experts involved, and ask critical questions when designing ligand libraries. Before diving into library design, let's briefly recap the virtual screening workflow. Library design involves selecting and filtering ligand libraries to maximize their relevance and quality for screening campaigns. The Schrödinger Therapeutics Group outlines 4 key stages of library design: literature review, goal confirmation, library selection and profiling, and filtering and labeling. Library design isn't limited to its use in preparing ligands for high-throughput virtual screening. It's also useful for building libraries for machine learning models, phenotypic screening, and lab-based screening methods. Much like baking a cake, while the recipe remains constant, the ingredients to build the library vary based on the project's needs. In this video, it's important to learn about the library design process so that later on we will be able to follow a project-specific library design request. Library design is a cyclic process. After the first cycle, screening goals may need to be realigned or the library expanded based on early results. Initial filtering is aggressive during the pilot screen. Later, new compounds can be added, such as those similar to known hits, or more complex molecules may be incorporated if their value outweighs synthetic challenges. Let's begin with the literature review, where ideally collaboration across disciplines ensures all relevant data is gathered and shared early in the project. This input helps shape key decisions in library design by leveraging both team expertise and broader research insights. Key stakeholders include medicinal, computational, and synthetic chemists, biologists, and project leaders. Typically, the biological target, a protein, receptor, or enzyme involved in the disease pathway, has already been established. Investigating whether the target has been studied before can reveal previously reported liabilities, such as toxicity or metabolic issues. Understanding why previous attempts failed, and in what stage of the development pipeline they failed, can help us approach the target, and specifically, the chemical space of the ligand, differently. In-depth searches in patents, the Protein Data Bank, and Chembl help identify existing compounds and IP concerns. Sometimes, the review uncovers opportunities for minor modifications to promising candidates to improve their ADME, administration, distribution, metabolism, and excretion properties. Although less common, synthetic challenges with a particular series can sometimes be anticipated before they become hits at the end of a virtual screen. Substructure or property filters can even exclude previously studied series or overly complex molecules from consideration. Typically, when using synthesizable vendor libraries as input for a screen, these filters aren't necessary. However, they become relevant when manually enumerating building blocks or performing de novo design. Computationally, 3D models of the target can guide precise ligand design by indicating where to perform linking or growing enumeration and identifying necessary protein-ligand interactions. Tools like Schrödinger's Ligand Designer are designed to help medicinal chemists leverage these models, making it easier to select or design compounds with a higher likelihood of binding. Published SAR, structure-activity relationships, can also be used to guide design through the selection of scaffolds and functional groups. And finally, it can be helpful to research the binding site size, charge requirements, potential competing binding sites, and the optimal pH for modeling to maintain consistency with lab-based parameters. For a screening campaign to succeed, all teams must align on these projects' goals. Essentially, what is the rationale for going after this target after the literature review, and what hits are likely to drive the project forward at the end of the virtual screen? Only compounds that would meet this criteria, if chosen, should be included in the ligand library. What qualifies as an acceptable hit can vary significantly between computational and medicinal chemists. The criteria below highlight some examples. It's the goal of the computational chemist to focus the screen to match the needs of the medicinal chemistry team as much as possible. For instance, requests like, "We need everything with a charge," are common, as these are often project requirements for medicinal chemists, especially when working with machine learning models. Closely tied to the literature review, it's essential to ask questions related to the likelihood of success. Will this hit identification method and library design strategy yield novel discoveries? Is this screening approach appropriate for my target? Are our design requirements too permissive or overly restrictive? Revisiting insights from the literature can provide valuable perspective, as these questions often need to be reassessed throughout the project. When selecting a library, it's important to consider its diversity, property space, compound availability, associated metadata, and size to make sure they meet the project's goals and resources. For instance, some vendors have target-specific databases, which may or may not be appropriate for your project. Let's skip to compound availability and reliability. Libraries are typically downloaded as SDF files from public databases like Zinc, PubChem, Enamine, and Chembl, which offer ready-to-screen compound libraries. Researchers may also purchase libraries from commercial vendors or design custom libraries in-house. When possible, utilizing an in-house inventory is often preferred for its immediate availability. Some libraries consist of virtual or theoretical compounds generated through reaction-based enumeration and combinatorial chemistry, which have yet to be synthesized. Others contain real compounds, synthesizable and commercially available, created using reliable reaction and building block combinations. Synthesizable ligand libraries are preferred because they yield actionable results, facilitating the transition to lab-based assays during the lead optimization stage of drug discovery. Attrition at the purchasing stage is common. Occasionally, compounds are expected to be available, but are not at the time of purchase. Several factors can slow down the purchase of ligands from chemical vendors, such as stock availability, custom or complex synthesis, purity issues, shipping and logistics, legal or regulatory requirements, and supply chain disruptions, among others. Unfortunately, even though these issues may seem minor in this course, they can add significant delays in the DMTA cycle down the line. Delays and disruptions can be minimized by working with a limited number of trusted vendors and ensuring that the latest version of the vendor library is downloaded before screening. Purchasing compounds from multiple vendors with differing processes can add complexity at the end of the screen, when resources would be best spent analyzing results. Tracking which compounds were actually purchased, retrieved, or assayed is essential, as initial buy lists of 200 to 300 compounds may yield only 100 or fewer available. Even fewer may ultimately be tested or confirmed, potentially skewing the hit rate significantly. Additionally, compound purity can vary across vendors, impacting results. Often, the vendor will provide an NMR spectra when they synthesize the compound for you to assess purity. Metadata is often available for download alongside ligands. It provides descriptive information about the data, including details such as how, when, and why it was created, who created it, and where it was created. Examples of ligand library metadata are shown on the right. Since metadata can vary by assay and source, standardizing file formats or cross-referencing libraries to remove duplicates and unify formatting can be valuable for future use. When incorporating structure files to modeling software, it's best to include the original SMILES from the vendor, pre-lig prep, to ensure a match with the compound source. At a minimum, upload and track the unique ID, such as the INCH key, to reliably trace the compound's origin. In some cases, the unique identifier may be a batch ID. Tracking only by title or variant is less reliable, as these can easily be modified or overlap with other datasets. SMILES cannot reliably be used to remove duplicates unless they were generated in the exact same way. Regardless of how well the library aligns with your project, filtering and labeling will still be necessary to further refine it. Filtering potential has been emphasized several times throughout the library design process. During this stage, property and substructure filters previously discussed are implemented to reduce the likelihood of false positive compounds or problematic hits. As a side note, while predicted properties can guide filtering, it's still essential to confirm these predictions with experimental data once the compound is available. The filtering stage is also an opportune moment to assess the team's risk tolerance one more time. For instance, does the team have a high tolerance for log D compounds? What predicted properties are the most valued? Is there a preference for compounds containing sulfur? Chemists often have differing preferences, and these decisions are important. The size of the library changes depending on the filters used. Sharing this information can help the project team reconsider certain properties. Pilot screens and validation runs, benchmark performance, and test filters, ligand libraries, and screening constraints, providing an opportunity to evaluate top-ranked compounds and refine the process. It's highly recommended to run one or more smaller pilot screens with representative ligand subsets from the full library before pursuing a larger-scale virtual screen. The length of the full screen and computational resource requirements can also be estimated from the pilot run. During a pilot screen, top hits are evaluated based on the desirability of chemotypes and the synthetic feasibility of the returned ligands. Among other attributes, you may choose to adjust your library composition, such as gradually incorporating compounds with known liabilities to broaden your region of chemical space. This approach is sometimes referred to as including bins from other collections. You may also choose to filter out unexpected and undesirable chemotypes that score highly. The pilot screen is also an ideal time to validate your model by calculating enrichment when known active ligands are included and labeled. Since pilot screens work with a much smaller subset of ligands than the full screen, diversity and novelty are expected to increase, which may make assessment of these properties during the pilot stage difficult. The number of ligands to screen during the pilot or model validation run is typically limited by resource availability. Pilot runs can range from a few hundred to tens of thousands of ligands screened, depending on library composition and model parameters. While the screening process itself can be relatively quick, investing time in a thorough library design upfront helps prevent redundant tasks and avoids the expense of unnecessary iterations. Thanks for watching.